It's my very great pleasure this morning to introduce our first speaker, Dr. Mark Maneri. Dr. Maneri is hopefully familiar to many, if not all of you, as the Helen Robertson Professor of Pediatrics. Dr. Maneri started his training at MIT and then went on to graduate from the WashU School of Medicine. And subsequently, he's devoted his career to a simple and profound goal of fixing malnutrition in African children. Along the way, Dr. Maneri has garnered many accolades and has produced some incredible and influential publications. But what I think makes him perfect as our first speaker for today for the Pediatric Research Retreat is another thing. You know, as pediatric physician scientists, I think we all dream of producing the kind of body of work that ultimately affects the health of children. And Dr. Maneri's research has done just that. He has used multidisciplinary techniques to really look at the basic pathophysiology and the optimal care of children with malnutrition, and along the way has truly saved countless lives. So I'm very excited that he's here today and in the country to speak. And a little birdie told me that we're also pleased to give him a happy birthday this morning. So happy birthday, Mark, and I look forward to hearing your talk on environmental enteropathy. It's my very great pleasure this morning to introduce our first speaker, Dr. Mark Maneri. Dr. Maneri is hopefully familiar to many, if not all of you, as the Helen Robertson Professor of Pediatrics. Dr. Maneri started his training at MIT and then went on to graduate from the WashU School of Medicine. And subsequently, he's devoted his career to a simple and profound goal of fixing malnutrition in African children. Along the way, Dr. Maneri has garnered many accolades and has produced some incredible and influential publications. But what I think makes him perfect as our first speaker for today for the Pediatric Research Retreat is another thing. You know, as pediatric physician scientists, I think we all dream of producing the kind of body of work that ultimately affects the health of children. And Dr. Maneri's research has done just that. He has used multidisciplinary techniques to really look at the basic pathophysiology and the optimal care of children with malnutrition, and along the way has truly saved countless lives. So I'm very excited that he's here today and in the country to speak. And a little birdie told me that we're also pleased to give him a happy birthday this morning. So happy birthday, Mark, and I look forward to hearing your talk on environmental enteropathy.
In here, cool. Yeah, the hardest part's always with these audiovisual things. But thank you so much, Audrey, for the kind introduction. And I do thank the organizing committee for inviting me to share this morning. And what I want to talk about is what I see I'm going to be spending a lot of time on in the next 5 years, which is the role of the gut and chronic inflammation in growth faltering in sub-Saharan Africa. So by way of background, I'll tell you some things, some preliminary work we've done in the last decade, and kind of what we're looking forward to. So when we talk about malnutrition in the public health community and the pediatric community, we're talking about growth faltering. And the Lancet Nutrition Series highlighted that stunting is child enemy number one. It affects 32% of all kids across the world. It accounts for 21% of DALYs in terms of children's disability. And it's not just being short physically, it's being weak physically, it's being cognitively stunted as well. So the short stature is a marker for lots of disabilities that children would carry throughout their entire life. Now, this picture here is as old as almost any of us, Nevin Scrimshaw, 1968. And it's been used to explain growth faltering for many decades. And the gist of it, I'm sure many of you are familiar with, is that children, particularly in the developing world, they get an infection, that they don't eat quite as well, their nutritional status is compromised, makes them more vulnerable to another infection, and what happens there is that growth is reduced. And there have been many actually very significant attempts to break that circle and show that we can have children growing normally. And so I'm just going to throw up a couple of examples. The bed nets, insecticide-treated bed nets, were about 10 years ago brought out in some very nice trials in Kenya. And these were the kind of trials where they were efficacy trials. People were going house to house to make sure people were using bed nets. They wanted to see how this reduced malaria transmission. And in fact, they could reduce malaria transmission by half or two-thirds, three-quarters. But they didn't really create, and that's the main infection these kids suffer from. And they couldn't really make children grow better by significantly reducing the most common infection they have. And then likewise, if you think about the other half of that circle, if you say, well, what about if we give people a perfect diet? I mean, all the nutrients they should need, meets every specification. And that's been done in a couple of instances. One time in Ghana recently, it didn't improve children's growth at all in a population that is chronically stunted. And a study that I participated in Malawi as well. I mean, we gave children what we thought was a perfect diet from 6 to 12 months, a perfect complementary food. All of these children were breastfed, and they didn't grow any better than the children that had no intervention and had porridge. So I guess where I'm going with these two anecdotes is that, this picture over here has got something wrong with it. I mean, if it could explain why children have growth faltering, then these kinds of interventions would be, front page news success stories, and we wouldn't be talking about why we need to do something about stunting, why in the Lancet Nutrition series in 2008 that remains enemy number one. So I've begun to think about this in the last 3 or 4 years in a different way, a different paradigm. It includes some of the basic things that Nevin put in. We have acute infections, and we need to manage our infections in such a way that our immune system controls them, but doesn't set up a chronic inflammatory state. We've got the gut sort of sitting in the middle of stuff because it's the agent of nutrient absorption. It's the most intimate interface between the immune system and our environment. And it's also a host to a variety of other organisms, our microbiome, our biome, and so forth. And then what goes in terms of nutrients and diet. And so if we want healthy growth from -9 to 24 months, the first 1,000 days, which has been kind of a mantra the last couple years, we're going to need to see that all three of these components are adequate and healthy. Now, what's environmental enteropathy? Well, it's something pretty nonspecific. If we look here at these histology slides, the top half is the normal small intestine. You can see the beautiful long interdigitated fingers. And down below is what the gut of probably 20% of 1-year-olds in the world looks like, and 30% of 2-year-olds, and 40% of 3-year-olds. Blunted villi, remarkably decreased surface area. If we stained this for presenting cells, we'd see there's excessive amounts of CD3, CD4, and CD8 cells. So a gut that isn't quite normal. These kids aren't sick. They're not telling you they have diarrhea or abdominal pain. They don't have anything that looks different about their stools. But they are not growing. So this is a very general term, so don't think of it as something like must be caused by agent X. It's probably got lots of causes because it's so common. It's very common in urban South Asia. It's very common in rural sub-Saharan Africa, which are different places. The things that we know about it is it's associated with poorer hygiene. So if you use less clean water or have access to less clean water, you're more likely to have this condition. And we know that we can recreate some histology like that without the inflammatory cells if we simply make mice micronutrient deficient. So it's probably related to your nutrition. It's probably related to some exposure in the environment. What does it do? I mean, this slide just basically tells you it reduces your nutrient absorption by about 5 or 10%. So it doesn't make you a grossly malabsorbing all your diet, but it cuts into it a little bit. And it creates a chronic inflammatory state, low level, which, and we know from all the different kinds of patients we take care of, is not conducive to growth. So one of the challenges with environmental enteropathy is that we can't see it very well. Like I say, the children don't look different. They're not complaining of anything in particular. We can't biopsy very many children. That's not practical and not ethical. So we use this surrogate sort of stress test of the gut, which is a site-specific sugar absorption test. And so this is where children drink non-metabolizable sugars like lactose and mannitol, and then we look for these sugars in the urine over a timed urine, complete urine collection. So the surface area of the gut is roughly proportional to the amount of monosaccharide that's absorbed. We do absorb some mannitol, and a predictable amount. And the disaccharide is proportional to the leakiness or integrity of the junctions of those cells, which is directly all about inflammation. So the lactose mannitol test, as I described, is what people are using in terms of research tool pretty much up to today to detect and assess enteropathy. It's cumbersome. You got to get a complete 3 to 5 hour urine collection, and remember our target age group's a 1-year-old, it's a 2-year-old, so this can have some challenges. If you use it in kids under 1, sometimes they get diarrhea. We all know we use lactose clinically for that particular effect. It's definitely an environmental thing. Peace Corps volunteers in rural sub-Saharan Africa have this condition because they tend to live in villages. And people that move from urban Asia or South Africa tend not to. It goes across the whole spectrum of the population, although you never see it in exclusively breastfed children. It appears sometime, can appear sometime in the first year of life, but is it present in adults as well, and kind of peaks at an age when children you might think would be less careful about their hygiene. So the presence of environmentalopathy as assessed by a lactose mannitol test has been associated for 20 years with growth failure. So this is some data from Gambia and Lund showing you basically a correlation between LM and linear and weight growth. And so this, to me, is a prime suspect as to why children are becoming stunted, is this chronic gut inflammation when it's not just about their diet or just about their exposure to other infections. We did some work here in the last 5 years or so, between 2005 and 2010, trying to see how in our populations that we work with are rural African kids today in the 21st century, what kind of role environmental enteropathy has using the LM test. This was one just an observational study we did, looked at about 450 kids, measured their LM and see how that compared with their linear growth. Is it looking like Lund? Lund's kids were little. They were, 6 to 15 months of age. And in fact, it is. And we had a great model to predict the change in height, and a lower LM or a lower lactulose excretion, which would be signs of a healthier gut, were associated with better growth. And other things you could imagine were as well. If you had a mother that took care of you, or if you didn't have HIV, you had more access to clean water, those kind of things. One of the things that people have said about environmentalopathy a lot in the last 30 years is, well, it's about eating contaminated food, and if you it's like getting too many bacteria in your food, and this is what's irritating their gut. So we took on a couple randomized double-blind placebo-controlled trials within the like around 2005 and then 2007 to try to see if we could intervene in that process to reduce small bowel bacterial overgrowth, presuming it might be there in populations of 3 to 5-year-olds. So, and the attractive notion of why this might be the case is you can create this in an animal model with dogs, an identical histology, by feeding dogs a diet that has more than 10 to the 4th coliforms per gram in it. One of the first trial we did was Lactobacillus GG, and a probiotic in a fairly large dose. These trials were done in such a way that we were sure the children were getting the intervention. We either had aides or students living in the village giving the children these interventions. So it wasn't a compliance issue. And our primary outcome change would be LM. And you can see on this slide that there's no difference. If you look at the bottom line there, 0.19 and 0.20 for placebo and lactobacillus GG means that this probiotic is not fixing EE to any extent whatsoever. And then we tried a non-absorbable antibiotic, rifaximin, which is used to treat small bowel bacterial overgrowth, sometimes by clinicians, and very broad spectrum. And again, this was a 7-day treatment where we had aides giving the children this antibiotic twice a day. So a true effectiveness study. And again, there's just really nothing to say here except that this doesn't change your LM score one bit. You can see in enrollment, rifaximin and placebo are really identical. So that has led me to believe that we need to look a little more broadly. We need to think that this may not be about bacteria. This may be about other kinds of agents, maybe about viruses, maybe about parasites. Certainly doesn't rule out for, ironclad that bacteria isn't involved in the enteropathy in these kids. And then we also said, well, okay, so they don't absorb 5 or 10% of their nutrients in general. Zinc is a nutrient that people talk a lot about. It's a key nutrient. I mean, experts would say that 1/3 of people in Malawi and nearly all of these children are probably zinc deficient. So we wondered if zinc homeostasis would be particularly affected by enteropathy. And that's because, if you might well know, that zinc is kind of a nutrient that we're especially vulnerable because we secrete lots of zinc into our gut. We secrete as much zinc as we eat in a day into our gut, and we need to be very efficient about reclaiming this zinc, or otherwise we'll fall into negative balance. So with zinc, it's not really just like putting it in and then the body takes up some of it. And they have diets that have lots of zinc in them, but also have lots of inhibitors to zinc absorption. So we looked at this in a group of about 25 kids. And we measured with 2 stable isotopes. We give one isotope IV, and we look for that in the stool, and that allows us to quantify how much they're losing, they're not reabsorbing. And we give another isotope orally, and we look for that in the urine, and that we can use that to calculate how much they're taking in from their diet. And they are taking in plenty, 25%. That might be what we're taking in here today of our diet. So that's not, even though they have lots of phytate, an inhibitor in their diet, they're able to absorb it. We wondered about this endogenous fecal zinc that then is lost in the stool, secreted into the gut. And we looked at that as our primary outcome and net zinc retention. And first of all, this population had 22 of the kids, 88% had enteropathy, so that's very common. The amount of endogenous fecal zinc these kids were losing every day was about 1.7 milligrams, which is huge. It's more than any of us probably, if we're healthy, are losing on any given day. Something's compromised about their ability to conserve zinc. And as you see here, on the x-axis it's LM, and a normal would be like 0.1, 0.15, and a elevated, the greater the number, the more enteropathy you have, the more affected you are of your gut. You can see these are biological observations, so you're not going to expect very sharp correlation coefficients, but endogenous fecal zinc is correlated with gut health, with more enteropathy, as more losses in the gut. And you see that these kids at a level of enteropathy that many kids have fall into negative zinc retention. So maybe this is an interplay with enteropathy and nutrition as well. So we came to, with those kinds of preliminary observations about a year ago, 2011, 2012, and we said, so what is really the deal with this? We've got all these big deal efforts going on to reduce stunting, to make children reduce growth faltering, make them grow normally. They're not working, whether they have to do with just giving nutrient supplements or fairly effective controls to reduce infectious exposure. We think it has to do with a neuropathy in the gut. It's a huge problem. It's one of the very biggest challenges that children face worldwide. It's a hard condition to detect and measure. There's no practical interventions to reduce it at this time. It's off the radar screen on most research agencies. So we're really not in a strong position. And we said, what do we need to do if we're going to fix growth, as Audrey alluded to, for kids in Africa and normalize growth, what do we need to do? So we need a better way to detect this. And with the help and encouragement of Dr. Tarr and his lab and many other people. So if I start mentioning people's names, then that's bad, because for every person I mention, at least I'm forgetting 20 to mention. So we have been looking at a new stool biomarker for EE. We looking at human mRNA in stool to imagine what that is. I mean, this is going to be mRNA that's coming from the upper small bowel. It's coming from the jejunum or duodenum. It's not going to be prevalent. It's not going to be most of the messenger RNA. Most of that's going to be bacteria, and if it's human, it's going to be from the colon. So it's looking for a needle in a haystack. We took about 450 kids and we measured their LMs. We took stool samples from them. We took blood samples from them, and we went about looking for some biomarkers. And we were interested that whether this human mRNA, a panel of them, because we knew that it could be more specific. Maybe we could look at things that were markers of inflammation. Maybe we could look at things that were markers of small bowel integrity and function. So that's what we endeavored upon about a year and a half ago. We had the notion that we should normalize the amount of mRNA there to a housekeeping gene, because we know that stool's not very homogeneous. Some stool might be more concentrated than others in the mRNA. We had to develop a couple of new techniques, and we were just very blessed that this came through really largely the efforts of some students who had very little lab experience that were working for me in a cubicle. But we knew that we were having some problems in the lab, so we sent these folks over there to work on testing and looking at different ways of isolating the mRNA. And ways have to be fairly conservative, because if we throw out half or two-thirds of it, we're losing too much of it. And we came upon a way using this NucleoSense EasyMag machine, bead-based technology, and lysis buffer. And this is, we could find this in these stools, these 450 stools, housekeeping genes present in every one of them. Whereas when you use a more conventional, RNA-easy technique, you can find it about half the time. So that was an important breakthrough. And then to detect these very low copy numbers, we needed something that could reliably tell the difference between 50 and 60 or 50 and 75 kind of things. And we heard about this new technology, I think about a little over a year ago, digital Drop Pcr. So you set up your PCR reaction, and then you emulsify these aqueous bubbles with the reaction in oil medium, and then you're detecting the fluorescence of a bubble. And since there's not very many copy numbers, you're probably detecting, one copy or something, and you can use an estimate of a Poisson distribution to count the copy numbers of several hundred accurately. And so we then thought about what transcripts might be the best guesses, and this is, of course, a strategy that's is limited by your imagination, and since my imagination can be quite limited, we may not have chosen the best ones, but we chose about 40 or 50, and as I said, normalized the copy numbers to a housekeeping gene. And 9 of these particular messages seem to be associated with enteropathy. And there you can see they are different Some of them are, they're listed there at the bottom: tight junction protein, Reg 4, Reg 3, or Reg 1b, some enzymes that just digest carbohydrate and protein, some molecules that are signaling the way T cells talk to each other, like toll-like receptor 4 or beta defensin. And then we turn to, well, I'll show you a couple other slides first that about this, a validation of this method, that if you have half as much, you get half as much signal. That's what this slide particularly shows. And that this detection technique is about twice as sensitive as a standard QRTC PCR technique. We turn to computational biology to try to predict enteropathy from this. We get some mathematical modeling, and we have some wonderful collaborators in California who've helped us with this. And they took these 9 probes and they said, which combination of these, can predict enteropathy? And we wanted, of course, the least number of probes as possible, but we also want the greatest sensitivity and specificity. 5 of the probes were chosen, and this part here is, for a person who really doesn't understand this in their heart, I'll try to explain it to you, but it's a larger polynomial equation that has up to 3 orders of magnitude with it, and you get these vast vector of coefficients, and then you optimize that with one database of several hundred samples, and then you can validate it with other databases that you have. Even though there's lots of coefficients, there's not many degrees of freedom, because you only have 5 mRNAs you're measuring, and you have a constant, so 6 degrees of freedom. And then you can write a computer program that works in a pretty public program like Excel, and just put your measurements in there, and it'll come out with the LM, a predicted LM, which we have now named 4M, mechanistic measure of mucosal malfunction. So in our model generation on the left there, that's pretty good. Measured LM versus this predicted. And what was really amazing to us is when we looked at this this calculated quantity, it correlates even better with these children's growth. So the model's made with the 2 things on the left, so you expect that to correlate, right? I mean, you're manipulating the coefficients and the powers and stuff, but to put it with a biologic, to get it to correlate with something biologic that you care about, these children's growth, was kind of encouraging to us. And then we validated it, and that validates about the same. These validated in kids allowing kids who are younger, which is more the target age group. They lived in a different geographic area, so it wasn't just we took some subset of the same group and then pulled it out and validated it, but it's a little bit of a stress test. And we're going to have the chance as we go forward with this work to try to validate this in a variety of other groups. So we think we're on the threshold of getting a new biomarker. We think we can do that with the detection of these human mRNAs in stool. We can do it very reliably. The folks in the lab can measure the same sample 10 times and get the same answer, so that's good. And then we went on very recently also just to try a couple of interventions, because we know we're going to, that's where it's all about. If you can deliver help, you're going to need to have to do something with an intervention. If we have a biomarker, that's great, because we can then measure the outcomes of these interventions, or we could get an idea of how prevalent or how much of a problem this is in a population. So we tried zinc and albendazole. These are things that are recommended by UNICEF and the WHO for everybody living in the developing world. Zinc being kind of a pharmacologic dose of oral zinc given after an episode of diarrhea. This effect of this zinc is not like adding zinc to your diet as a nutrient. I mean, it's some kind of pharmacologic effect on cell junctions. And the albendazole, of course, is a deworming medicine for parasites. And we did this before with zinc down in the corner with LM is our outcome on the y-axis. And you can see 3 groups, zinc, placebo, and albendazole. And in a situation where enteropathy was increasing, gut health was deteriorating, these interventions, given one time, 1 dose of albendazole or 1 course of zinc, and then measuring it a couple of months later so that to see what effect is there when the gut has a chance to regenerate and heal, and not the immediate effect of that medication. Those are statistically different, and we look forward to seeing what that 4M marker is in these kids as well. So as I come to a conclusion here, probably I have plenty of time for questions. How might we heal the gut and get children growing? Well, we can do something simple like promoting the access to clean water, reducing the exposure to coliforms. There's some data from Brazil suggesting dietary glutamine might be useful, so incorporating foods with more glutamine in it. Micronutrient supplementation and fish oil, and I think probably next year at the research retreat you'll at least see a poster from Hannah Smith, who did a double-blind placebo-controlled trial of those interventions recently. We don't have the results yet to tell you, but fish oil can be used to reduce inflammatory status of the gut mucosa in inflammatory bowel disease. And then maybe even promoting the consumption of something like lactoferrin and lysozyme. So these are proteins that we all know are in breast milk in very large quantities. I mean, lactoferrin is like over half of the protein in breast milk. And they have an effect in laboratories and in mice and in cell culture assays of promoting normal gut tight junctions and gut cell functions. And we also can get these in fairly large quantities because some friends of mine have made transgenic rice that expresses lots of this protein. So this is something you could just grow like rice and add the rice powder to the top of their porridge to start with, or introduce the rice into their diet. So it's not just sort of a pharmacologic outer space kind of intervention, but something that might actually be able to be used. So those are some of the hypotheses we're going to be pursuing in the next few years with our new biomarker. We're going to look at our biomarker in terms of other populations, folks in South Asia who have come from an urban area, which probably have environmental neuropathy from a different stimulus, a different basis. So maybe they need a different panel of RNAs to predict their enteropathy. And the consequences of this maybe 10-year adventure could be lower stunting, better growth and development, less clinical malnutrition, and better child health overall.